Welcome to my poster where we evaluated experimental factors that can influence measured IC50 on NaV1.5 recorded from high and medium throughput automated patch clamp devices. In this study we used two automated patch clamp devices, the patch liner and the SynchroPatch 3-4i. Due to variability of IC50s obtained on different APC devices during the CIPA study, we set out to find out which parameters could most influence IC50 values on NaV1.5. In Figure 1, we compared data of NaV1.5 obtained using different voltage protocols. Regardless of protocol used, the recordings were stable with no rundown in vehicle. The protocol used did, however, affect IC50, which you might expect for compounds, for example shown here, clozapine, that preferentially block the inactivated state of the receptor. We next looked at whether cultured cells or frozen stocks of cells influenced IC50 values. We found that the IC50s of 24 compounds were well correlated with a Pearson's correlation coefficient R² of 0.989 when using cultured or frozen stocks of cells. We also found good correlation between IC50s obtained on the patch liner and the SynchroPatch 3-4i for 11 compounds. The late current is an important component of NaV1.5 and can be recorded either at the end of the -50 mV step, known as the persistent current, or during the ramp phase of the voltage protocol shown here in Figure 3 at the top. This is known as the late current. We found that measuring either the persistent or late current yielded similar IC50s for ranolazine. Temperature has been shown to influence biophysical properties and IC50 values of voltage-gated ion channels. We found that temperature had no effect on the Vhalf of activation, but did induce a statistically significant shift of 7 mV to more hyperpolarized potentials at physiological temperature versus room temperature. In addition, temperature caused a twofold shift to higher potency of mexiletine. We found that incubation time influences IC50 of compounds blocking NaV1.5, shown here on the poster on Figure 5. We found that for 4 out of 5 compounds, 5 minutes of incubation in compound was sufficient to reach steady state, but for particularly sticky compounds such as tafenidine, a longer incubation time was required. However, when experimental parameters are standardized, IC50s are stable across different plates and different days on the SynchroPatch. Here lidocaine is shown where the concentration response curves for different days and on different plates overlay almost exactly. And indeed, IC50s of 18 compounds acting on peak NaV1.5 current measured using the SynchroPatch 3-4i compared well with the literature. To conclude, we found that voltage protocol does influence IC50 values and would recommend the CIPA set ramp protocol, which enables effects on both peak and late current to be measured. Temperature effects, biophysical properties and pharmacology of NaV1.5, so temperature must be controlled, and a minimum compound incubation time of 5 minutes is recommended. It is critical to standardize protocols for automated patch clamp so that IC50s can be compared across sites, platforms and with the literature. If you have any questions, please join me on Tuesday the 13th of July to discuss the poster in more detail, or write me an email, ali@nanion.de. Thanks for listening and enjoy the rest of Physiology 2021.